Okay, can you hear me and see me?
Yes, we can. Thank you.
All right. Well, it's a pleasure to be here today. Thanks, Emmanuel, for inviting me, and I'm really excited about being a participant in this meeting. So I'm going to launch right in. Okay, wait a second. I'm not moving through my slides. Okay. So I have 4 goals. I'm going to make sure we're all on the same page with respect to understanding the hibernation phenotype, and it's in quotes because it's variable depending on the species. I'll be talking specifically about circannual hibernators, or ones that follow a strict circannual rhythm, and our model organism is the 13-line ground squirrel. My thesis is that mimicry of natural hibernation has a lot more promise for safe, reversible space torpor versus current medical practice. And then I'm going to try and argue that natural hibernation can translate to space torpor in humans using several lines of argument. And then talk to you a little bit about how to get there. And I think in the introduction, Emmanuel made a very good point that understanding model organisms and the genes that are involved is going to be the best way. And so we need very precise understanding of bona fide hibernation in a real mammalian hibernator. Okay. The hibernation phenotype in 13-line ground squirrels and other small-bodied mammals like them, which hibernate on a very strict circannual or about 1-year basis, includes core body temperature, which can approach or even go slightly below the freezing temperature of water, heartbeats that are ranging between 3 and 10 beats per minute from their normal resting heart rates of around 200 beats a minute. So this is a profound depression. And of course, the profound depression of metabolic rate from 2 to 3% of basal. Now, this is a very selected group of physiological parameters that are associated with a state that we call torpor. And what's really important in these circannual hibernators is that torpor is discontinuous. So what you see here is a body temperature trace over most of a year, starting in August, September, October, and so forth. You can see that this period of torpor is restricted to about half the year. And in fact, it's punctuated by periodic arousals back to a euthermic condition where body temperature resumes at near 37 degrees Celsius. After spending about 1 to 3 weeks in torpor, the animal spontaneously and repeatedly rewarms.
So first of all,
yeah. Somebody's not muted. Okay. So they spend about 12 hours in this period of rewarming. I'm going to argue that in the case of these animals, this is a handle for us to better understand the genes that are involved in orchestrating this torpor arousal cycle. And moreover, it's important to realize that this winter heterothermy is quite different than summer, where the animal behaves like a proper mammal. So we have also a transition between these two states that give us a handle for finding genes that could be important for orchestrating and surviving the torpid phase. Okay, science fiction has long been interested in the idea of using torpor in space travel, but for me, at least, the concept of applications of hibernation for space kind of transcended that science fiction realm and moved into what should be science realm when I saw this paper from Spaceworks that was presented at an Astrophysical Society meeting. And they had actually designed a transport vehicle for 96 people in torpor. These 2 modules each hold 42, anyway, 96 total people in torpor with 4 people tending them in another module of about the same size. And this sort of illustrates the advantages of torpor, because if you don't have to feed and cleanse the environment from the people maintaining high metabolic rate, if you can get them into low metabolic rate, their demands for travel are far less and more people can be packed into a small space. So I thought this was pretty amazing, but found that they wanted to do this by elaborating current medical practice, in particular these 2 features, therapeutic hypothermia and total parenteral nutrition, both of which are fairly invasive. And in this figure taken from a paper that I published with my co-author, who will be speaking later and has a much better grasp on all the medical aspects of the problems of therapeutic hypothermia and TPN, we sort of argued that this is so invasive, it's going to be problematic. It's not really going to transport anybody successfully to Mars. And instead, we should look to the natural hibernators for an understanding of how they can get such profound metabolic depression. It's reversible and it happens without harm, so that when they emerge in the spring, all these organ systems that would be challenged by a therapeutic hypothermia approach, as it is now practiced in medicine, are simply not a problem, and the animal resumes its normal activities for summer without harm from that long period of essentially cold storage and reperfusion. So, okay, well, that's crazy. Can people hibernate? Well, no, they can't exactly right now, but I want to make the argument that if we understood how hibernation worked in a natural model, we should be able to achieve this state in humans. And so my first argument is that hibernation really sits on the extreme end of a continuum that we already exploit. So every night we enter slow wave sleep. It's a metabolically depressed state with a slightly reduced body temperature. If we elaborate that, I mean, just the slow wave sleep shows this. This is 8 days, and to be honest, this is a ground squirrel temperature trace during the active season. But what you can see is this regular dip in body temperature each night. And of course, the correlates of that are a lowered metabolic rate, heart rate, respiratory rate. And that's a state that we all enjoy on a daily basis. If we take it bigger and deeper, we get a state called daily torpor. And the example here is an edible dormouse who's in a state of daily torpor. And you can see really the key thing, which is true also of hibernators, is that metabolic rate is depressed before body temperature. So they lower metabolic demand first. Body temperature follows because there's less metabolism producing heat to make the body temperature warm. And then to rewarm, metabolic rate is activated again and the animal begins to rewarm. And in this case, the pattern is daily. But in the case of hibernators, now the pattern is extended. So the period of torpor extends from 1 to 3 weeks with these spontaneous arousals back to euthermia. And in each case, metabolic rate is dropped, then body temperature follows. So you take the demand down, you're not asking for as much oxygen delivery, and then cooling just further exacerbates the energy savings that you get from this strategy. Okay, so argument number 2 is a phylogenetic argument, and that is that the idea of using torpor, whether it's in a daily pattern or a multi-day pattern like hibernation, is really quite widespread among mammals. And so here what you're looking at is a phylogenetic tree showing lots of different species. Their species and genus names are in different colors, so you can see the different groups of mammals that are represented. But at the end of each of these branches, there's a little blue bar or a little red bar. And the blue bars are true hibernators, and the red bars are animals that use daily torpor. And you can see these are interspersed on this phylogenetic tree and quite broadly represented among species of mammals. So if we look at this a little bit in a more pruned way, where it's easier to see the main points, what we find is that all 3 of the main groups of mammals, monotremes, marsupials, and eutherians, like humans, have pluses and minuses on the ends of these twigs. And these are species that do or do not hibernate. And since those are widespread among all these groups, the simplest interpretation of this phylogenetic tree is that the common ancestor of all mammals was a hibernator. And that this animal, of course, our genes should be then present in all the extant species. These asterisks are representing deep hibernators, or animals that use this near freezing type temperature that you saw for the ground squirrel pattern earlier. And you can see, again, these are very widespread on this phylogenetic tree, representing each of the 3 main groups and 3 of the 4 main groups of the eutherians. And it may be that we just don't, there are a lot of species of mammals who've not been studied well enough to know if many more species are capable of this as well. So my argument is that phylogenetically, we should have the genetic hardware that it takes to hibernate. And maybe a few of those genes have been, over evolutionary time with the disuse have been lost, but nevertheless, if we understood the mechanism, we should be able to, by and large, recapitulate the phenotype. Okay, the third argument is that humans do not necessarily die when they become cold or lower their metabolic rate. And actually, people who work on human cell culture freeze cells repeatedly and they come back from that. So our cells are not intrinsically unable with protection to survive extreme hypothermia or metabolic rate depression. But moreover, people themselves can be resuscitated from extremely low metabolic rates and low body temperature. And so there are a couple of really famous examples in the medical literature, and Dr. Nardin will talk about this more later as well. But we have here 2 papers that are credible where human body temperatures went below 14 degrees Celsius and people were revived with no long-term ill effects. Now, that's not to say that their revival was a bit too heroic for a daily or for a trip to Mars, but at least in the short run, it's not intrinsically impossible to survive low body temperature or low metabolic rate for a human. Excuse me. So we've been working in my lab under the hypothesis for a long period of time that the hibernation phenotype is determined by differential expression of common mammalian genes based on that phylogenetic argument. And the way we envision it is that these circannual hibernators take both environmental signals and an endogenous circannual rhythm, which definitely has to do with reproductive hormones, as many people have shown. Those converge on the brain, we think modify gene expression such that cells throughout the body also are affected and instructed to modify their gene expression, and taken together, this is the hibernation phenotype. So we really arrive at this question, which genes are important? And the answer is the differentially expressed genes. And yes, I see this is somewhat of a circular argument and maybe not a bona fide hypothesis, but this has been driving our work for a long period of time. And I think I have some pretty good evidence that it's a fruitful approach, which I hope to convince you of now. Okay, but let's just think about it a little bit abstractly at the beginning. So let's say we have a seasonal gene that's really important for survival of heterothermia. And you can imagine that this animal doesn't need genes that protect it from, let's say, that ischemia reperfusion that they undergo every time they rewarm rapidly after the 2 or 3 weeks of cold. But they might need that as extra protection during the season of heterothermy, so they only express it during that season. And so the expectation is that we would look for differential gene expression between animals in the heterothermic phase and animals in the homeothermic phase. And likewise, if we want to ask what's going on in the torpor arousal cycle, what drives that need to rewarm, and then what takes the animal back into torpor. One of the things that we learned early on is that both transcription and translation are cold sensitive in a hibernating ground squirrel, just as they are in a mouse or a rat. So they don't work. If the animal has gene products that are essential for orchestrating torpor, it's going to need to make them during those warm periods. And so you could imagine that it enters torpor with a high level of that gene product, and that over time, the gene product decays such that it can no longer maintain torpor. The animal rewarms and it makes more, now it can resume torpor. It's a really simple idea, but I think it's valid and worth exploring. And the one thing that should be immediately obvious to you is that if you sample randomly across this wedge or this wedge, you will get the same answer, and you will never know that that gene is differentially expressed and required for torpor. So those averages would be equal. And I'm going to argue now that it's really important that you know the exact timing of the animal in torpor. How long has it been torpid? Is it just 1 day in, or is it most of the way done and about to come out? Likewise, it's really important not to just take an aroused hibernator, but to know exactly where in this arousal process it is. And that's what we've done by using precisely monitored animals, taking body temperature variation as the way to determine where the animal is in its torpor arousal cycle. So we actually we have these minimeters, which were battery operated. Batteries could fail by the end of spring, so we got really compulsive and implanted also a data logger. We can't recover the data in this until we recover the device, but this one sends a signal in real time to tell us where the animal is in the torpor arousal cycle. So these get abdominally implanted. The animal's allowed to recover and then moved into a hibernaculum. We were really lucky at our campus because this cold room is an outer cold room. It had a sign on it, don't go in. And in the inner cold room where it's really quiet, the animals were hibernating, not disturbed for months at a time. And we could monitor them remotely with their implanted telemetry devices to collect a tissue bank at very precise time points in the hibernation phenotype. So in this study that I'm going to share with you today, we did RNA-seq to find differentially expressed genes in 3 brain regions. we have these states. We have from the homeothermic period, we have summer, so getting ready to hibernate, but not too close. These are early August animals, having finished hibernation and spontaneously decided to stop in the spring. And then we have these 4 states from the hibernation period itself, taken from the period where the phenotype is quite stable. So the bouts of torpor are stereotypically about the same for the bulk of the season, but they're shorter on both ends. These were taken from the stable part of the season, where we have animals that had just been warm for 2 to 3 hours, animals that had started re-entering torpor based on a body temperature between 23 and 27 degrees Celsius, late torpor animals that were 85% of the previous bout's length. So again, with taking advantage of the stereotypic length of these torpor bouts, and then arousing between 7 and 13 degrees Celsius. So we knew pretty precisely where these animals were. And I want to argue that we have essentially 2 cycles going on here. We have the seasonal cycle, which we could represent in this way, where IBA animals become the proxy for the warm state in the winter season. And then the animals entering torpor, going to late torpor arousing. This cycle is the torpor arousal cycle during hibernation. And again, we have 3 brain regions. We chose the cerebrum because it's fairly quiescent during torpor arousal cycles versus the hypothalamus and the medulla, which both have work to do to kind of organize the cycles themselves. We had 90 samples for RNA-seq. Each of these 6 states was represented by 5 individuals from each brain region. And excuse me, what you can see is that we were surveying about 13,000 genes. So we quantified transcripts based at the gene level. And most of them were expressed in all 3 brain regions. But if we looked for differentially expressed genes, with really quite a high bar for calling them differentially expressed, we find that only about 20% are shared among the 3 brain regions, and the rest are somewhat specific to the 3 different brain regions. But in every case, we separated fairly easily, so I'll focus from now on hypothalamus, the hibernators, except for IBA hibernators, but here you can see a nice line separating all the hibernators from the spring and summer homeotherms, where IBA was standing out as a very different state. And a different way to look at this is by looking at the pairwise differences, just simply counting. If we look at a spring animal and compare it to a summer animal, we find that 28 genes are increased in summer, but 39 are relatively higher in spring. So only about 70 genes change in this transition. Likewise, in the transitions across the torpor arousal cycle, the number of gene expression changes are very small. But what you can see are this large number around comparisons to IBA. So those 2 to 3 hours just after rewarming. There's a seasonal change, and then there's a torpor arousal cycle change. And both of these are quite large, indicating a big burst of transcription during the IBA. So another way that we looked at these was to cluster them based on phenotype. And we found that only really 3 features mattered. One feature that matters is what season is it? Is it a winter season, i.e. a hibernator, or a spring season? And, or I'm sorry, a homeotherm or a heterotherm, basically. But we also found that body temperature matters. So in this data set, for the first time actually in any hibernation data set, we're really separating the differences that tell the animal I'm a winter hibernator versus a summer active animal. And we're separating the differences that occur because of the cold in the torpor arousal cycle. And both of these are quite dominant in the data. So what you can see also is that there are more changes in the genes that are depressed in the cold than there are in those that are increased in the cold. But still quite a lot of them are increased in the cold. And if you remember, I told you transcription stopped in the cold. So you might wonder how that happens. And there's a clue when we look at the actual gene expression data in more depth. So these are now individual genes, not the summary of the class, but the individual genes. And I just want to highlight a few points here. One is that we see a very prominent indication of RNA binding proteins. They're seasonally expressed, so they're elevated in winter. They're also elevated in the animals, not in the IBA, but actually as animals literally prepare for entering torpor. So these are the if you can look up here, the middle 4 classes are the hibernators, and then we have the the homeotherms spanning on either end. But you can see this elevation in entrance. So this is something that happens late during the warm period, that poly-A binding proteins are coming up later. And then there's also a big burst of them that are just unique for the IBA itself. And so what this tells us is there's a tremendous control at the level of RNA stability and management of the RNA populations, RNA turnover and stability, as opposed to transcription. But we do see a signature for transcription, and that's common with IBA animals and the spring and summer animals. So there's an elevation of transcription very early after the animals recover their body temperature in that torpor arousal cycle. So I want to remind you that when you use RNA-seq to get data about differential gene expression, you are measuring steady state RNA, and you don't know if you're seeing increased transcription or decreased degradation if an RNA accumulates in this population. But our data does give us some indication of that, because we see a big burst of transcription factors in the IBA state compared to all other states. So we know that early on there's an elevation of transcription. We have found evidence for, as I showed you, transcription changes, but also stability, additional polyadenylation. We have published a story about RNA modifications where adenosines are changed to inosines. And there are also variants of splicing and 3' end processing in these data. So there's a lot to go through. But I guess the idea I want to leave you with is that a special subset of RNAs accumulates during this long period of torpor, such that it reaches its highest level as animals are beginning to arouse. Those transcripts are specifically stabilized, and that selects a program that initiates as soon as the animal's warm enough to start transcription and translation again. So this by itself could reset a timing mechanism, and I'm really interested in exploring that idea further. So I just want to end with a caution that much of what we do in studying hibernation, if it's driven by hypotheses that come from species that don't hibernate, is like this guy looking for his keys under the light because he can see there, even though he lost his keys over here. So here's our hibernator. And he's not revealing the secrets with this approach at all. Instead, we need to study the hibernator itself to understand hibernation. And with that, I just want to thank my collaborators, particularly the bioinformaticists that have helped me with this large data set, Austin Gillen and Ray Few, and then many other people over the years who helped with the collection of this remarkable tissue bank, which I think needs to be studied in far more detail than it has thus far been studied. Thanks very much.
Thank you, Dr. Martin, for this great presentation. And we have now 5 minutes for Q&A, so I'm going to ask Dr. Stringman to help us with the questions.
Sure. So the first question is more methodological. Was the torpor induced along with true seasonal change, or was it inducted in the lab despite the season? And
no, it was the
mechanism between states?
Yeah. So the way these animals were mainly purchased from the Anschutz breeding facility for 13-line ground squirrels. Not all of them, but most of them. And we brought them into our labs in usually late July, early August. Gave them a few weeks to recover, did the implant surgeries for the 2 devices to measure body temperature, gave them another couple of weeks to recover from that, 2 to 3 weeks. And then as the animals begin to exhibit torpor, which you can see their behavior changes, they start building nests. So we give them some nestlets and they start building nests and they look lethargic. And pretty much around the beginning of October, we'd move them into the cold room. And as long as they, within a week, started exhibiting torpor arousal cycles, we'd leave them in there. And so it's sort of natural. It's not completely natural, but pretty good.
Has any sort of a follow-on to that and related, has anyone studied the role of the circadian clock or circannual clock, if there is such a thing, in hibernation?
Yes, and there's a fair bit known about that. So there are some experiments that have been done in the past few decades where people lesion the suprachiasmatic nucleus, for example. And it disturbs the pattern of hibernation, but it doesn't take away the ability to do it. Some people have looked at hibernation genes. I think some of those data sets are a little bit circumspect because The animals were kept in light dark cycles because maybe sometimes animal husbandry, we were so lucky because our animal facility said, okay, we believe your telemeters, you can monitor your animals remotely. We could leave them in constant darkness. They never had a light dark cycle. No one went in at 10 a.m. every day to give them cues. And I've seen papers where even just compressors running at regular intervals would give animals cues for timing. So I think you have to be really careful for that. But certainly the lesioning experiments tell us that the SCN is not absolutely required for hibernation, but it is required for their timing. And there's also another really nice study with the pineal and melatonin. And what that study showed is that the melatonin rhythms provide information about the length of day, and that's really important for setting the circadian timing of hibernation. If you allow the animals to free run in either constant light or constant dark, at least sciurids, it's been done with chipmunks, marmots, and ground squirrels, the bouts of torpor, the season of hibernation initiates earlier every year. So the free running clock shifts forward. Instead of being 12 months, it would be 11.
Right. Great. Thank you. Another question is, in your opinion, how would a size difference factor into this, say squirrels versus humans?
Yeah, I mean, that's why people like bears as models for human hibernation, because they're more about the same body size and they get a pretty strong metabolic depression without much body temperature depression. So, I mean, it takes more energy to warm a larger body from a cold temperature than it does a small body from a cold temperature. So, yeah, I'm not necessarily advocating that we should try and achieve freezing in humans. I could see the benefit. You wouldn't have to warm the capsule quite so much. You know, it could save even more energy. But I don't necessarily believe that you have to do that to have a tremendous energetic savings and a benefit from a torpid state. Yeah, does that get far enough into your question?
Yeah, that's fine. I'll ask one last one and then we'll move on. How do you envision translating this to a therapeutic approach inducing torpor in humans? That's a big one and may need to be pushed to the discussion. But
yeah, I mean, I think we have to understand better what's really important. It's not just going to be gene expression. It's going to be metabolites. There's a whole slew of things that are important. I don't, you know, you're probably aware of the recent papers that have shown it's possible to get a hibernation-like state by modifying a specific neuropeptide, for example, in a certain region of the brain. I don't think that's the whole story, but it might be enough.
Well, we have a whole bunch of other questions, but I think we need to move on, and so we'll save the remaining questions for the discussion period later. Is that
okay? Thank you. I'll stop sharing my screen.
Thank you, Dr. Strengman. Thank you, Dr. Martin, for the really good presentation.